Hello and welcome to my latest blog. Today we're going to be talking about the plain language summary of a recent paper that we published on the use of metagenomics for One Health surveillance of environmental antimicrobial resistance in a UK veterinary centre. This is a plain language summary of a paper that so myself and colleagues across UCL and the Royal Veterinary College wrote, which describes how we use sequencing to help identify what clinically relevant species and antimicrobial resistance genes might be present in a veterinary hospital setting. In this paper, we specifically looked at the use of metagenomics, or the study of multiple different organisms within a sample. I should say that whilst I've used the terms clinical and hospital here, I mean them in a veterinary sense. The two worlds of human and animal medicine rarely cross over, and so they both use these same terms. At the Centre for Clinical Microbiology, we naturally work very closely with the infectious disease teams in local human hospitals, and scientists and clinicians of both the human and animal variety are becoming more and more aware that we need to link up and share our knowledge, as no sphere exists in isolation. And the linking of human, animal, and environmental research is known as One Health and is becoming increasingly important. As part of our work with Pandora IDNet, the Centre for Clinical Microbiology also works with the Royal Veterinary College and other more animal-focused sites to undertake One Health projects and provide capacity development to others looking to do the same. The human clinicians that we work with knew that I've been working on sequencing protocols for various pathogens, and between us, we decided we'd see if we could translate this sequencing knowledge into the veterinary world. As an aside, when I was going through this data and the previous literature and seeing how fairly gross our pets might be, although contrary to popular belief, dogs' mouths are similar to humans' mouths when it comes to the number of bacteria they contain, it didn't actually put me off sort of interacting with my dog. While she's not the most disgusting dog to ever exist, she doesn't roll in or eat poo, for instance, nor does she lick me in the eyeballs like some dogs I could mention. She's still got pretty funky breath, as she's 10 years old now. Millions of people clearly feel the same way, that the benefits of owning a furry friend outweigh the potential gross negatives. There are loads of studies showing the health benefits we get from owning pets, such as reducing our stress, most of the time, helping us stay fit and active, and reducing feelings of loneliness. Just follow your vet's advice to keep illnesses to a minimum. So, back to the paper. Until recently, the focus of antimicrobial resistance has been mainly in human patients and clinical settings. Multi-drug resistant organisms that commonly cause healthcare associated infections in humans are increasingly being isolated from animals in the environment. These include methicillin resistant Staphylococcus aureus, or MRSA, and E. coli. Much of the research focusing on AMR in animals looks at those animals involved in the food chain. After all, livestock animals make up 62% of all the biomass on Earth, nearly twice that of humans. Much less research has been conducted on the animals that we live more closely with, but pet ownership is growing rapidly. 1/3 of households in the UK own a dog, and over a quarter own a cat, and this has risen since the COVID-19 pandemic, of course. Even in countries where pets aren't traditionally common, many people still live alongside animals within their households, such as guard dogs or stray animals that live within communities, and of course those kept for meat and other produce. While there's not been much research into the roles these animals play in AMR transmission, scientists are starting to consider dogs, cats, and other small animals to be AMR reservoirs. A bit like human hospitals, veterinary hospitals are thought to act as reservoirs for multi-drug resistant organisms, as many, often of course sick animals, pass through and share the space. Most previous literature looking at multi-drug resistant organisms and AMR transmission in veterinary environments have utilised phenotypic testing, so using agar plates and antibiotic discs, to identify species and genes. Sequencing, where the DNA of organisms is looked at, can be used to provide a much clearer understanding of multi-drug resistant organisms, as it can link these organisms, so look at the transmission, identify resistance genes, which make organisms resistant to the drugs we use to treat them with, and look at the molecular tools they use to share these genes, such as plasmids. In the UK, whilst there is a national clinical AMR surveillance programme, there is no surveillance programme nationally for AMR in companion animals, and only limited data exists. In lower middle income countries such as those in Africa, there is virtually no consideration for the role that animals and in the environment play in the spread of AMR. There are currently very few standardised tools or guidelines for genomic surveillance of One Health AMR. So, this project aimed to utilise metagenomics to identify what AMR genes and multi drug resistant organisms might be present in a small animal hospital environment and compare them with those seen from clinical or phenotypic speciation and antibiotic sensitivity tests. We went to the veterinary hospital armed with a big box full of swabs, sponges, and pipettes. In fact, it all came with me on the tube. We collected swabs and wastewater samples from various sites around the hospital. Some examples include from the dog kennel waste drain, in the sink U bends, and from the animals' consulting tables. And all the samples were preserved in a liquid called DNA RNA shield, so that, as the name suggests, the DNA didn't get broken down whilst it was being transported back to the lab. Luckily, this time not on public transport. Because we wanted to look at all of the species within the samples, we extracted the DNA directly from those samples, so from the wastewater or from the preservation liquid that the swabs were kept in. When we prepared a sequencing library, so we had to amplify the DNA using PCR and then attach barcodes to each sample so that we could run multiple samples together and still know which sequencing reads belonged to which sample. We then sequenced our prepared samples using the Oxford Nanopore MinION, and this can take up to 72 hours. And typically, each run, with around 20 samples on, can kick out up to 300 gigabytes of data. That's the equivalent to roughly 30 2-hour HD movies or 150,000 photos. We then analysed the sequencing data to look for AMR genes, plasmid, and clinically relevant pathogen species. Our sequencing data was then compared to phenotypic antibiotic sensitivity testing and speciation of infections that the veterinary hospital had collected from patients and sent off to their laboratory over the previous year, so we could see if there were similarities between the two data sets. We found a number of interesting things in our data. Firstly, when we analysed the phenotypic data from the veterinary laboratory, we found a high proportion of phenotypic resistance to the beta lactams, especially in staphylococcal infections. And beta lactams are a large class of antibiotics that are commonly used to treat infections. When we looked at our genetic data, we found lots of antimicrobial resistance genes in the veterinary hospital environment that can cause resistance to these beta lactams. The most common resistance genes that we identified in the veterinary environment were APH, which causes aminoglycoside resistance, SUL, causing sulfonamide resistance, BLA-CARB and BLA-TEM, causing beta-lactam resistance, and TET, causing tetracycline resistance. And these are all commonly used antibiotics in companion animal welfare. So it is a little bit worrying, although not surprising as these genes are very common. When we looked at what areas in the veterinary hospital harboured the most number of resistance genes, these were found to be the preparation rooms where pets are prepared for surgery and have minor procedures, the dog ward, the data bay, where staff input information onto computers, and the consultation rooms, where pets and their owners go to meet the vets. When we looked at what specific sample types had the most resistance genes, we found that the kennel waste drain had the greatest number, followed by the consulting tables, bins, sink plug holes, and door handles. We looked at what plasmids were identified in the samples and found that 24 gram-positive plasmids were identified, mostly associated with staphylococci, and 4 enterobacterial plasmids were identified, encoding carbapenem and quinolone resistance genes. These plasmids may be the key to how resistance genes move within the environment and also between living hosts. Finally, when we checked the sequencing data and compared it with the phenotypic data to see what pathogens we could find, 64% of species found to be the cause of infections were also identified in our metagenomic samples. Pseudomonas aeruginosa and E. coli were common infection isolates that were also commonly identified in the environmental samples. Our study identified AMR genes, plasmid, and multi-drug resistant organisms of relevance to both human and animal medicine in metagenomic samples from the veterinary hospital. Areas of the hospital where a lot of animal handling happens, such as the preparation rooms and consultation rooms, as well as areas in which dogs were present, were more likely to harbour AMR genes, plasmids, and multi-drug resistant organisms. It's important to note, though, that many of these pathogenic species, such as E. coli and Pseudomonas aeruginosa, are commonly found in lots of environments and are not necessarily going to cause disease. Context and further metadata are often needed to fully understand how these organisms move about. When considering our results in the context of infection prevention and control measures, or how the veterinary hospital might go about reducing the spread of multi-drug resistant organisms and AMR genes, more comprehensive disinfection of certain areas, especially surfaces that come into contact with animals or animal fluids, such as the consultation tables and bins, may reduce the number of potentially harmful bacteria present. While this was quite a small scale project, known as a pilot project, to see what we might find, we're using this knowledge to develop grant applications for larger, more comprehensive projects. We've got a number of collaborators in Africa who are interested in using our sequencing pipeline to look at antimicrobial resistance and pathogenic species in their food chains. And examples include abattoirs as their common reservoir, and lots of animals pass through these areas.